Afternoon. Sorry. Okay, so let's get started. Welcome to, this is our second keynote talk for the showcase. And my pleasure to welcome our speaker today, Dr. Shankar Subramaniam. So, a couple of words I wanted to sort of say about the showcase broadly, which I brought up last night at the Honor Academy, College of Beauty Honor Academy induction ceremony. One of the things that we do with the showcase is really try to use it to foreshadow and amplify other efforts that are happening. And so last year, our showcase was really focused on AI, and we'll have to see some of the culmination of that tonight as we launch in this very room the new AI Center as part of the ICC. This year's showcase is focused on convergence. And convergence is clearly a word, we need to come up with a new word, we tend to overuse that word, but the idea of solving grand challenge problems that require expertise from multiple disciplines is critical to how we're going to move forward and is a major priority for this university. And the showcase is meant to sort of help tell that story and move us along in that journey. And at some point we will have, Michelle, join us up here. The new Center for Convergence and Innovation, which will be a building built around research that is occurring in the cracks between different disciplines. Okay, and as we started thinking about convergence, we were brainstorming on who to invite as speakers. And no one came to mind to me faster than Shankar Subramaniam. Shankar is currently a distinguished professor of bioengineering and computer science and engineering and cellular molecular medicine and nanoengineering at UCSD. He has broad research interests that all sort of occur at the interface between computing, engineering, and medicine, and navigates that continuum harmoniously throughout everything he does. He currently holds the John and Irwin Jacobs Endowed Chair in Bioengineering at UCSD. He is the founding director of the Bioinformatics program at UCSD and was chair of bioengineering at UCSD for 5 years until 2013. Prior to moving to UCSD, he was at the University of Illinois at Urbana-Champaign, where he had a long list of faculty appointments that I won't go through, which is a little longer than what he had at UCSD. He's a fellow of the IEEE. He's a fellow of IMB, so that's the International Academy of Medical and Biological Engineering, and a fellow of the AAAS. He won the Genome Technology All-Star Award. He's won the UCSD Faculty Excellence Award, Research Award, and he's a distinguished scientist at San Diego Center for Supercomputing. San Diego Center for Supercomputing. He's on the external advisory boards for several bioengineering programs across the country. Kind of small, maybe you've never heard of them, universities like Johns Hopkins, Case Western, Penn, Rice, UT Austin. So none of them of the quality of Michigan Tech, but not slouches. He's currently an overseas advisor for the Department of Biotechnology, the government of India. And sort of the least noteworthy of all of these, he was actually my PhD advisor. So it's been particularly exciting to have Shankar here. Shankar, thank you.
Thank you so much, Dennis, for his kind words, and he's been very kind. In fact, I learned more from him before I left Urbana-Champaign, and the one thing I learned from him is that I need to be more organized than I ever was, and that was really something he taught me. Those of you who know him know he's an exceptionally organized individual, and that's a phenomenal thing. And thank you, Dennis, for inviting me. It's really a pleasure to be here. I've never been to UP, as you call it. It's my first visit to UP, and I'm really overjoyed that you brought the San Diego weather here for these 3 days, for me. And I was really delighted, and I was worried a little bit about the weather, and so it turns out that it is fantastic. Thank you again. And I enjoyed meeting a lot of folks over the last past 2 days. Amazing energy, talent, and enthusiasm for the future, and it's a privilege for me to be here. And thank you very much. I thought I'll talk. I had a very, very good, precise instructions from Dennis on what to talk about, and I always follow instructions. And so he told me that I shouldn't give a very serious life science talk, or I work in multiple areas of systems medicine, system biology, and the interface of receiving data and computation and so forth. He said, no, I want to hear about kind of a semi-high level talk about the kinds of things that can be done. So I followed the example. I do have a little of examples from my own work, from our own work, but in addition to that, I just thought I'll give you a perspective of where things are going in the future. So this is where I am. Dennis forgot to mention to you that I really love to have feedback and input. And if you have any questions during the process of presentation, I don't need to complete all my slides. So if you have any questions or suggestions or ideas for me, I would be grateful to you. I'd be delighted to hear from you. Okay, so I just wanted to start by saying, so what if an intelligent alien lands on planet Earth, and this intelligent alien has sensors and which can gather bits of information, and let's say the alien comes across the following. The top one is kind of artwork, and the bottom one is something you will recognize because it's all in and they are huskies. And so let's say he looks at that, and what will he or she, we don't even know, or it, whatever it is, the alien, would make out in terms of measuring zillions and zillions of bits of information. How will they decipher this? So this is where, I mean, we are in the form of aliens today because we are measuring things which are so multi-scale, large scale. I'll show you 2 examples. That is not clear to us how we are going to decipher and make sense of these. This is the landscape I want to draw for you as to where we are going. In fact, big data and the future of AI, it's not as if we are saying that it may happen or it's a part of our life. It's going to be an integral part of our life, and it's going to be one of the strongest futures where we are going. And I'm going to illustrate this to you as we go along. And in fact, I want to give you the intellectual viewpoint of AI. I prepared this because I was challenged by my chancellor to prepare a proposal for creating a new school of computing, information, and data sciences, which started 2 weeks ago. We are approved all the way up to the University of California, Office of the President. So I said, here's what are the intellectual challenges. You have data, big data, and then obvious question is how do we even structure this data, represent this data? How are we going to deal with the complexities associated with the data? How are we going to process this data? How are we going to encrypt this data? How are we going to, use this data towards analysis of this data? So this kind of provides the intellectual landscape of the entire field of data sciences. And in fact, when I prepared this for the proposal, the University of California Office of the President got it reviewed by experts across the country, and they said this provides a really good landscape for where the data is going. So I will highlight some of these areas here during my talk, and I'll tell you what are the kinds of things that are forthcoming as we go along. So the largest challenges for data science are very, very large data. How do we structure this data? How do we organize this data? How do we process this data? How do you analyze this data? How do you reduce the data? So this is a common problem because contrary to the popular opinion, you can take everything and put it into TensorFlow from Google and out comes knowledge is a complete myth. I don't think it ever happens. So therefore, this is really where the organization and the processing is coming forward. The dimensionality of data is the largest problem that we have, and I'll illustrate through examples. How do we reduce dimensions? It's called the curse of dimensionality. I'll show you how we reduce dimensions. And I'm also fond of saying that context-free data processing is content-free data processing, because if you don't have the context, then you're not going to get any meaningful results out of it. And then causality is, to me, personally, causality is one of the biggest challenges in data, because we can look at correlation. Correlation is easy to do, but causality is a much more complex issue, and you need time series data, and you need longitudinal data, and then that gives you temporal causality, and not necessarily always mechanistic causality. So I'm going to illustrate through examples again how we deal with this. I will dive into the problem. I want to give you a highlight view of big data. So this is Meerkat radio telescope in South Africa, where you have, the goal of this Meerkat look at the 1 kilometer array is to really gather data for looking at the evolution of galaxies and the evolution of the universe. That's the goal. Every second, the L-band digitizer samples 1,712 samples. The amount of data generated is a petabyte of data in a day. And that's, if you didn't realize what it was, it's equivalent to 500 billion pages of standard written text. So clearly, I mean, we're not going to be able to read through or look at 500 billion pages of standard text. So this is definitely a problem for AI. And what do you want to get at the end of that? At the end of that, you want to look at the evolution of galaxies and the formation of this big picture of galaxies. And in fact, that this is not a myth. This particular array discovered a supermassive black hole, Sagittarius A, which sits at the center of Milky Way galaxy. I mean, you could argue, why do you care? We care because if you want to look at the evolution of the universe, in fact, if you want to look at how the universe evolved across time, this kind of an array is able to really build a picture of how the universe evolved over a period of time. So I just want to give you the largest of data and how do we convert this data. To give you an example from cosmology, I'm very fond of looking at the sky, so this really provides us a perspective on how much data is there these days. Let me go to the area that I'm more comfortable and familiar with, which is the human genome. I'm sure most of you have, or many of you have used this facility called 23andMe. I'm not plugging for 23andMe, please don't. I have no stocks in 23andMe. However, you can sequence your genome for as little as $100 or $125. And it gives you a fantastic insight. Most people look at it for ancestry. They say, I come from 13% of me comes from Poland or something like that. I'm not worried about that. You can actually get your genome sequence and do your own analysis of that sequence. And you can look at, for example, whether there is a propensity for Alzheimer's disease or Parkinson's disease or things like that. But these are very simple examples. And in fact, simple examples because you know that one mutation is causing this, and so therefore you can figure out if that mutation exists or not. But the more real thing is that we have variations. If you take across any two human beings, there are several million mutations which differentiate people. Despite the fact we are all 99.999% similar, we still have 3 billion base pairs. We have millions of base pairs which are distinct. And so how is the sequencing thing going? In fact, if you look at the amount of just data, let's talk about data for sequencing. We are already into the zettabyte regime, which is 10 to the 21 bytes of data. We are going to be into the yottabyte regime. For those of you who don't know what these bytes are, we go from deca to kilo to mega to giga to tera to peta to exa to zetta to yotta. So we are already into zettabytes of data. And even these zettabytes of data, we still will not decipher human diseases. I'll tell you why. In fact, we did a back-to-back calculation on the dimensionality problem. Don't worry about the math because I'm not worried about the math. The math is still a straight point here. So even if you sequence 10,000 genes or 100,000 genes and you have millions of mutations, we are going to simply consider a linear model. Let's not even worry about a non-linear complex reality, but let's consider a linear model. You're going to end up getting just a matrix which is infinite, which means that you're not going to be able to reduce the dimensions to find all the phenotypes that are associated. So this is just to give you a glimpse of the fact that it's very complex and how you reduce the dimensions of data. So I'm going to walk you through an example on this. Now, the next few minutes, maybe 20 minutes, I'm going to focus on dimension reduction, data reduction, and how we extract knowledge from this data reduction. That's what I'm going to do. So the dimensionality is the curse of dimension. In fact, the first person to define the curse of dimensions was a mathematician called Richard Bellman. And Richard Bellman said that the number of samples needed to estimate an arbitrary function with a given level of accuracy grows exponentially with respect to the number of input variables. This is the dimensionality of the problem. So he coined the word the curse of dimensionality. And so this is now one of the fundamental motifs of AI, and machine learning and deep learning. How do you deal with the curse of dimensionality? In fact, if you go to many AI conferences, they're centered around the curse of dimensionality. How do you deal with it? How do you convert higher dimensional data into lower dimensions, yet preserve the data integrity? The question is, you can always say that I'm going to take all this data and eliminate 90% of it and look at 10% of it. That doesn't make sense because you don't maintain the integrity of the data. So how do we maintain the integrity of the data? I'm going to give you a couple of examples which illustrate this. If you take a digital human, imagine, and this is not George Orwell's fiction, this is real. In the next 50 years, we'll walk into a hospital or a clinician's stay. We'll go in and there'll be a machine which will go into like Woody Allen's famous film Sleeper, and then you'll end up coming up with millions of measurements on a human, and it'll tell you the state of your health to multiple degrees. This is going to be a reality 50 years from now. I may not live to see it, but on the other hand, it's going to be a reality. So how do we deal with such data? So the living system, for example, is multi-scale. This is one of the fundamental issues when we think of data reduction. We have genes, nucleic acids, proteins, cells, tissues, organs, and human beings. And these are all the modalities. Simple. I'm just giving you a few examples from one of the reviews. These are all the modalities by which you make measurements. There are many, many more modalities, but these are some simple modalities. And let's look at the scales. The scale is really the most exciting thing, spatial and temporal scales. If you look at spatial scale, you have 10 to the 9 orders of magnitude. You look at a person is 1 meter, let's say 1 meter long, maybe much more, 2 meters. The electrical length scale of a cardiac tissue is 1 millimeter. Cardiac sarcomere is 1 micrometer. And pore diameter in a protein is 1 nanometer. And then time scale wise, you have 15 orders of magnitude. Even in simple scales, in human lifetime, let's say 70 years, protein turnover is 10 days. The smallest thing is the Brownian motion of things, and that's the microsecond, so you have 15 orders of magnitude. How do we deal with this? The diversity of models we need to generate, or all of these models. This is pre-AI. So what do we do with these models? You have networks and statistical models, stochastic models. What is missing here is AI and machine learning models. And this is what I want to focus on for the next few years. So we had a very interesting project that NIH launched, and NIH had this I was chairing this panel which dealt with this large project. The project was very simple. It said, okay, we got about 50 different types of cancer. We've got, hundreds of drugs that have been FDA approved. What drug should a physician use? Should they use one drug that is better for one person, another drug is better for another person? So they decided to do an exhaustive analysis of this landscape of 50 by 100 by multiple measurements, types of these things. They measured genes, they measured proteins, they measured chromatin, which is the one that defines the landscape of the cell. And so they measured all of these things. And they said, how do we make sense from these measurements? So they said, okay, how do we get the fingerprints? So we'll take, for example, a graduate student in my lab, who's now an assistant professor at WashU. She took all these data together and tried to figure out what is the best modality for us. So now I'm going to convince you one size doesn't fit all. So it's not just one AI method or one deep learning method. Context becomes very relevant. So what she did was she said, okay, I want to say take histone structure and drugs. I've got these 50 drugs that I want to figure out how many minimal modes do I have that will be sufficient for me to describe this landscape. So she used a method called non-negative matrix factorization, because that turns out to be, we work a lot with analytics, and so that turns out to be a very interesting method. Okay, she used this method, and then this is not very difficult. Taking large dimensions, she reduced it into 4 dimensions or something like that. And these 4 dimensions are these dimensions where she said, these are the 4, don't worry about the details, because I want to give you an idea. These are the 4 dimensions of the landscape dimensions, which connect drugs to the chromatin. Okay, so it's sufficient. Even though we have 50 drugs or 100 drugs, these 4 dimensions cover what the cell does or the tissue does. What about proteins? So the protein landscape, she said, it's not a protein network. They are much more diverse, much more complex. So she decided to use a different method, partial least squares reduction method. And then she said, okay, I'm going to reduce this and then show, this is a very straightforward mathematical method. She used it to show that here's all the methods. I result in 10 possibilities. And then don't worry about the details again, the 10 possibilities she did. She said, I'm going to combine all of this to produce a model. So she combined all of these
to produce a very interesting and complex model. Don't worry about this model. This model gets reduced. When I convert this model into a model that biologists and cancer specialists understand, it comes down to a model like this. So this model tells you that if I start with a drug, a given drug, the drug will go through this pathway, it will go through this mechanism, and this mechanism is what is causing the drug to be functional or non-functional. So for an individualized way, for a given drug, for a class of human beings, I can now use this network to ask the question, is this model a good model? Okay, so there's one example to show you how you can reduce data dimensions using knowledge towards understanding cancer biology. I'll give you one more example. In fact, in recent times, one of the fascinating methods, and this didn't come from biology or life sciences, this method is called embedding methods for representing and semi-supervised. What do embedding methods do? Imagine I have a 20-dimensional data. Now, I can think of 2 dimensions. I can probably think about 3 dimensions, visualize them. 20 dimensions, there's no hope for visualizing these dimensions. How do I deal with this? So there's a beautiful method that is developed by mathematicians. It's called embedding methods. What the embedding method does is that it computes a graphical representation of the data set in high dimensions, reduces it to lower dimensions, and calculates the equivalent of some kind of Euclidean distance. It can be whatever metric you use, and it reduces this complex space into a simple graphical representation in 2 dimensions. And the original method was called T-distributed stochastic neighborhood embedding, TIST. But don't worry about the name, but the basic idea is that you are reducing dimensions by embedding it into lower and lower and lower dimensions, till you get to a point where your lower dimensional embedding represents truthfully or faithfully the higher dimensional data. Okay? And so if it doesn't make sense, please stop me. I'm happy to go over it in detail. So this was a fantastic method because the moment that we discovered that this method was applicable, people started making use of this method for various contexts. Here's an example of dimensionality reduction. Take large dimensional data and reduce it. Disney, for example, reduces it to a clusters, and UMAP, which is a better representation, preserves a larger cluster distance, breaks it down into larger clusters. Now we can ask the question, okay, I've reduced the dimension. What does it mean? What does this reduced dimension mean? Does it even have anything that you can interpret? Does it have something that can be physically meaningful? I'm going to show you a really interesting example where the interpretation of this. So COVID times, in fact, COVID, all of you know about the pandemic. What COVID does is the SARS-CoV-2, virus, CoV-2 virus. It binds to ACE receptor in epithelial cells and starts an immune response. The immune response is sometimes good, sometimes it's not sufficient, so you get severe infection or mild infection and so forth. This is a standard COVID picture taken from a scientific American level viewpoint. So when COVID initially came, we asked the question, people started measuring large amounts of data from blood. They took blood from patients. And we were very fortunate that I had, both my lab and other labs were able to get a large number of patients and get their blood samples. So we asked a simple question, can I take this high-dimensional space where we measure RNA, single-cell RNA sequencing from these patients' blood, this is plasma plus cells, measure them. Can we make sense of what happens in severe COVID, what happens in mild COVID? Very simple question. The question is simple, but the reduction of the question into knowledge is where the complexity comes. So we took data sets, which are many, many data sets. We got all this, we're all collaborators, so we got all this large number of data sets. We said, okay, I want to really convert this data set into knowledge. There are 80 cell types, so one of my postdocs, she worked very hard to identify all the cell types in an immune system. These are all the 80 cells in the immune system. So we got markers for every cell, we got markers for every cell type. And we said, okay, let me take this large dimensional space, reduce it using UMAP, and this is from COVID map, and this is healthy map. What does it tell you? What it tells you is that it breaks down the cluster into cell types. So now I know I can look at the cell heterogeneity in a severe COVID patient and cell heterogeneity in a mild COVID patient. Okay? Why is it useful? It's useful because I've taken this vast amounts of patients, large number of patients, we had thousands of patients. We took thousands of patients, took their blood measurements, converted, reduced it into the cell types which are present in healthy and in severe COVID patients. Why is it useful? Because then I can use a drug, therapeutics, which will help in terms of reducing the severe COVID patient into the less severe COVID patient. Right? And so we did this, and then we were able to map this very beautifully into looking at patients. We were the very first ones to look at the immune system at this large scale. And we were able to show what cell types are more in a COVID patient. For example, we were able to show that there's myeloid cells and lymphoid cells. Lymphoid cells are all the T cells and B cells, and all those myeloid cells are macrophages, monocytes, neutrophils. So we're able to show what happens in severe COVID patients that you can address, all using this technology for dimension and data reduction, right? And number of genes in each case is about 20,000 genes. There are thousands of patients. It's 1,000 patients times 20,000 genes times 80 cell types. I mean, and the times severe patients versus non-severe patients, right? So it's a large dimensional problem. So this UMAP approach, this embedding approach, helped us to reduce it. And we were able to come up with very simple rules saying that this is what happens in a severe patient, this is what happens in a mild patient, this is what is true in a healthy patient. And so in fact, there are companies who started developing drugs to address this question in a more direct way. This was very early, this we published in fact in 2020 or so, or early 21, right when COVID came. Okay, so this is one example of complex dimension reduction in a large space. Let me give you, this is the map of what we did. It shows the myeloid and lymphoid, how these cells change. And I don't want to go into, this is not a biology talk, I don't want to go into details, but this is kind of the things you can represent, how to reduce, zillions of bytes of data into simple models, which will tell you something useful. At the end of the day, you want to translate it into something beneficial. So next, we said, okay, autoencoders became very famous. Most of you know what autoencoders are. Autoencoders, one other way of doing data and dimension reduction. So we said, okay, autoencoder is a special type of neural network that is trained to copy, input into its output. Here's a very good example of an autoencoder. This is a published paper actually with somebody. They took a very crude version of Mona Lisa painting, and then they encoded it into a latent space representation. And the latent space representation was transformed into a real space representation into Mona Lisa. So this was a real proof of concept of autoencoders. I don't remember whether it was Google or one of the companies, but this was one of the publishers of autoencoders. So how do autoencoders work? Why is it useful? So we asked the question, how do we use this autoencoder method? So we said, okay, here's all the breast cancer data from across patients. The 4 types of breast cancer. There's luminal A, luminal B, triple negative, and HER2 positive. So can I feed all this data into an autoencoder network? We developed an autoencoder network specifically for this, and show whether when a breast cancer patient goes to a hospital and you do an assay, can you say a priori this patient is luminal A and requires this treatment, is luminal B requires this treatment. And we were very successful in showing that it's able to predict with 90% accuracy, between 85 and 90% accuracy, the type of breast cancer that is present in your disease. I'm sure if you have more data, we can get more and more accurate. So it's a question of data richness. So this is another example of how you can reduce things. Next thing we said was, now I was very excited by this paper I saw. We said, data has a shape, data has a topology, and the data's shape has information. Most of the times when you reduce data, you lose information about the shape of the data. Right? We have no concept of the shape of the data when we reduce the dimensions. For example, I do principal component analysis. I lose every information about piece of information about data. Okay, I do embedding methods. I still don't have information about the data. So when a professor from Stanford, who's also a founder of a company, came to us and said, hey, I have a method for for using the shape and the topology of the data. I said, fantastic, so let's do it. So we then took this idea. The idea is very simple. The idea is to take an input, which is time series data, to embed it into, examine the shape of the data, And then do topological data analysis, which means look at the topology of the data, not just simply look at the data number, amount, dimension, but look at the topology. And then you see you get beautiful patterns of the shape of the data. So we said, why not use the shape of the data, embed it, and find out. So he said, great. So I looked at a paper. In the meantime, while I was thinking about this very hard, I came across a paper. It's not what from my lab, it's what from. So imagine we all go to sleep, we have resting states of the brain. Is it really resting? What is the brain circuit like? It's very useful because it predicts how healthy we are, what diseases we have, what types of things, how is our day going to be. Think about a scenario in the future where when you sleep, you're automatically sensed in your brain and it tells you next day, these are the best things you can do. For example, like tennis, you can play ice hockey in the afternoon at lunchtime, it will be better for you, things like that. So therefore you want to have ability for doing this. So these people did some fantastic stuff. They took a lot of patients. They made them healthy humans. They want to see the resting state of the brain. So they put them, when they went to sleep, they put them to MRI. They measured every half an hour, or every 10, 15 minutes, something like that. They measured the MRI brain state continuously. And then for 2.5 hours or 3 hours, and then they did this repeatedly many times. And so they had vast amounts of data. They were able to take this data, use the topology and the shape of the brain to map it into figuring out what regions of the brain are activated when you're in a resting state. So when you're in a resting state, what gets activated? They were able to beautifully map this across different parts of the brain. So I'll show you that, I can skip this, I want to show you this. What they were able to show was that when they mapped it to different regions of the brain, they were able to look at functional connectivity in adults who are doing exercise and adults who are not doing the right kind of exercise. They said, who has got more propensity for Alzheimer's disease? They were able to really do this to look at the risk in terms of the neural connectivity from the resting state of the brain. Again, this is not science fiction. This is reality in coming 2 decades, 3 decades, or 4 decades. We are going to see more and more of this, right? So I thought it's a beautiful example. So we then said, why don't we try it in the case of breast cancer? And we learned something new. And indeed, we came across something very interesting, because when we use this topological data analysis for breast cancer data, we discovered a new subgroup of this type of cancer. And when I went to my cancer specialist, or a few cancer specialists, and told them the subgroup, they said, we kind of wondered why our patients were not subject to good treatment with these subgroup. So because this was HER2 rich, they were mistaking them for HER2 positive cancer patients, the luminal patients. So now, many of my breast cancer collaborators, we work on breast cancer as one of the areas of research, our collaborators are very excited about the ability to decipher, and this is all done by using data and this topological data analysis method, right? So it is really powerful. How we can reduce data towards translating it for human health is becoming really crucial, and I'm very excited about this idea. Let me digress and talk about time series and causality. I'm really interested in causality because correlations are useful, but they don't give you as much information as causality. Yes, there is. Say again.
Who you get into this?
Yeah. Yeah.
So take PCA for now.
Yeah.
PCA is going to identify where there's maximum variability. The algorithm is going to do what the algorithm is designed to do. And if we think about trying to understand the essential dynamics of an elephant walking across the room, it's going to start by that. We can subtract out those privileges. But what we're interested in is the tail moving. We might give you the head moving. Right? So the algorithm is designed not based on the problem we're trying to solve, but how we're trying to do that dimensionality reduction. And whenever we have these issues, we solve them all the time. We're talking about essential dynamics, proteins, ice, energy, and you're talking about. You often lose the thing that we're interested in because it's incompatible with what the algorithm, what computer scientists are mathematicians and stuff. How do you ensure that there's coherence?
Yeah, Dennis raises a beautiful problem. So, I mean, in words of computer scientists, you talk about, first of all, PCA is a linear method, so let's not even worry about it. But We talk about unsupervised learning versus supervised learning. A large amount of unsupervised learning may give you not the context of what you're looking at. Because when you take your data and do principal component analysis or nonlinear discriminant analysis, whatever method you use, you're assuming that the data has some inherent properties that you're using it to really divide space into some functions. Now, the inherent property of linear, nonlinear may even be a component of projection of the data, which is why we strongly believe in supervised approaches towards learning. When I say supervised, it doesn't mean that I'm going to give the answer I want to the method, but I want to introduce constraints into the learning. So all the methods I showed you today so far as examples of constrained approach towards learning. Even when they did the analysis of the radio telescope, the array data, they had a structure of the universe in mind in terms of looking at the constraints and reconstructing. Right, so these are not, they are inputs into the system towards understanding the structure of data. I hope I kind of addressed your question. So most of the methods, when I talk about causality, the same idea comes into picture. Your elephant example is a good example because I'm going to give you the context of the elephant here. So, most methods for time series and causality have an intrinsic idea. So here's the idea. So, we know this from mechanics and from dynamics and so forth. Because when you say you have an equation of motion, which tells you that if I'm here at this point in my automobile and I have this acceleration, I'm going to be at point X. That's a very simple example of causality. With large dimensional data, causality becomes much more complex because you don't have an intrinsic understanding of the structure of causality. So what does causality do? If I have time series data, then I can do all these kinds of things. I can do power demand, forecasting for calls, I can do inventory forecasting, supply chain forecasting, maintenance forecasting, all these, all the forecasting methods, all the weather predictions, everything were based on some notion of causality. What is the best way to look at causality? So we have been examining this for now several years, for over a decade. And so we came up with an idea that autoregressive approaches towards causality is really very good. What's the idea of autoregressive approach? Suppose I have time series data. I have time series data on time t, t minus 1, t minus 2, t minus 3, etc. And I have multiple measurements. I'm going to have a matrix which says that I'm going to try to figure out at what point in time do I have no causality, no effect on the outcome. That's what the autoregression idea is. That's the whole idea of autoregression. For example, I have yt is a function of t minus 1, t minus 2, t minus 3, etc. And I have a whole matrix or series of such equations. Can I then use the causal derivations? And causality is a very interesting, again, in response to some of the questions you raised. Causality has multiple ways of thinking about it. Norbert Wiener, and I suspect Tim is going to talk about Wiener later in the discussion today. And so Norbert Wiener, for example, he said for two simultaneously measured signals, if you can predict the first signal better by using the past information of the second signal, then we call the second signal causal of the first one. Very simple idea, but Wiener actually used it in his cybernetic idea. And so the Granger was the one who actually, I spelled it wrong, Granger. So Granger, who was actually at the University of Illinois, he was the one who really was the one who discovered this causality time series approach. And he said, I'm going to use an autoregressive approach towards causality. So he said, okay, can I use this in complex data sets? Yes. Can I use it, for instance, people are using it for brain measurements. They look at time series data and brain electrode measurements. And they showed how you can get which parts of our brains are interconnected from this Granger idea. So this Granger's notion of causality said, okay, which parts of the brain circuitry is connected. Today, in fact, a lot of papers, if you go to Journal of Neuroscience or go to Society for Neuroscience, there are many, many papers on using Granger causality because there are neural circuits. This is the basic idea of the regressive model. Again, don't go into details. There are these k times k autoregressive coefficient matrices, and we can then combine them with learning methods. In fact, in my lab, we have used them with other deep learning methods in order to really derive time series. So here's an example we applied. We said, okay, here's magnified cell, here's all the measurements you make. Can you predict the pathway through which the cell communicates the signal from outside to inside the cell and to come up with a phenotype? So we used combination of these data sets using autoregressive models, and Mariam Masnadi Shirazi in my lab, she's a graduate student, and she's electrical engineering graduate student. And so you can imagine students, cross-fertilization students. She used this data from the cells in order to build a causal model, time-varying model, where you can look at it this time, 0 to 4 minutes is what happens, 3 to 7 minutes is what happens, 6 to 10 minutes. How do we know this is true? We go to experiment and we can validate. We did validate it when we publish it. So here's an example of an engineering approach to version and a simple model. Here's another example she did. She published a paper on cell cycle. She said she was bold enough to say, I'm going to take the data on a cell and then I'm going to predict the cycle of the cell without knowing anything else. Without telling a priori that this is the phase of the cell cycle, this is one phase, another phase, third phase, fourth phase. People know this, but we didn't know this. We didn't in a mammalian cell. So she was able to predict accurately using greater causality the exact time period of each cycle, member of the cell cycle. Again, I'm not giving this example to you to illustrate what we did, but to show you that time series data can help us infer causality using the right approaches. So here is an go on to do this. Here's a second example, forecasting. Systems which are really, deterministic, great, you can use greater causality. What about a chaotic dynamical system? On the left, this is traffic in Calcutta, in India. And if you don't think it is chaotic, then there's something wrong. And so, it is entirely chaotic. That is a real, that's taken from a real image photograph. And on the right side, I have this beautiful, there's a guy called Conway. He invented this game of life. The game of life idea is very simple. You take any pattern you want, dots connected to each other, dots. If you have 2 neighbors, you die. If you have 3 neighbors, you increase by 1. If you have 4 neighbors, you status quo. If you have more than 4 neighbors, again you die. So if you evolve this pattern in time, it's unpredictable. It's sort of rules. Your rules of the game. It's in fact called rules of the Conway's rules of the game of life. This is how you want to study evolution across society. So this is really pseudo-chaotic system because you don't know what the result is going to be. In fact, if you like it, go to Google and search for a game of life. You will get a game that you can play on your computer and generate patterns, and you'll see you will never predict exactly what pattern you're going to end up with. It's really a beautiful game. So this is exactly how do you deal with problems like traffic in Calcutta or the game of life. So we decided to look into this because many biological systems have these intrinsic properties. And so we said, okay, how do we look at this chaotic? So I asked this electrical engineering student to say, okay, let's look at forecasting approaches in chaotic dynamical systems. So we came up with this method called, okay, here's one pattern, here's the second best reconstruction. I have n reconstructions of my chaotic behavior. The n can be very large, it can be, a million. From these n reconstructions, can I come up with a model that will be able to predict at a given time point what is going to happen? Right? And it's not that complicated. So we found out we used the radial basis function network, non-parametric forecasting approach in this dynamical system. And we are able to predict with very good accuracy a number of known examples in real-life systems of evolution. Behavior for honeybee, for example. And so things like that. And so we're able to show that. The very last part of what I want to tell you is dimension collection, feature selection. This is supervised process. You really want to have, in fact, all of AI is now, many of you know this, are going to be given by constrained AI, feature-based AI, AI which is knowledge-driven AI. So these are all words for the contextual AI. These are all words for looking at how do we build in some features into the AI in order to learn. So for example, you want to remove irrelevant features, it's easier to debug, easier to understand, easier to build, make training faster, increase the performance of the model. That's the whole goal of what we want to do with AI. So how do we do this feature selection? I'll give you an example. So we had a very interesting story years ago. So we looked at people get vaccinated. All of you or most of us get vaccinated for flu every season. And sometimes the flu vaccine changes, but we get vaccinated. As we grow older, the vaccines are less effective in elderly patients than in younger patients. In younger patients, it's more effective. In older patients, 65 and older people, it becomes less and less effective as we grow older. So one of the goals we decided to look at was that we worked with the Vaccine Institute in Emory, CDC in Emory, and with Emory University. We said, how about if you get longitudinal data on measurements on vaccination? Measurement of post-vaccination, we take patients, we recruit patients to have each year, we look at patients and measure their blood levels at time 0, 2 days, 5 days, 7 days, 15 days, etc. We measure the blood. And let's ask two questions. One is, across years, are there common mechanisms that are triggered across vaccination, or there are distinct mechanisms? Simple question. Second, we said, okay, why don't elderly people not respond as effectively as younger people? So these are the two questions we wanted to ask. So we said, okay, no worries, we got this data, beautiful data sets from Emily, and we are able to really take all these different data types, transcriptomics, metabolomics, cytokines, cell populations, and we're able to feed this into, this is the study design, let me skip that. We're able to use ensemble classifiers and committee of networks and input features. We took a lot of features and input them. And then we did a multi-layer perceptron. Most of you know what multi-layer perceptron is, a neural network with multiple layers. And we trained it on this in patients. And then we introduced feature space to reduce these dimensions. And then this is the workflow. Again, I skipped that workflow. We have to show across seasons, for example, 2008, 9, 10, 7, 8, 9, 10, etc. The patterns were very similar. That means the vaccines responded in a similar way to flu vaccine. Independent of what vaccine we used, it responded similarly, which gave confidence that the type of approaches people are taking towards vaccination makes sense. Okay, what about elderly and young? So when you look at module based in elderly and young subjects, there's a lot of differences. The young people, in fact, had more inflammation. Older people had more inflammation, which made it difficult for them to respond. In fact, we had published this paper where we showed in older patients there's increased pro-inflammatory status, which means we are all more inflammation in our body when you get older and as you get older. In fact, today it's called, the word that's used is called inflammation, which means you're aging with aging, your inflammation goes on. So how do we deal with this? So we told our collaborators, why don't you give these elderly patients ibuprofen or Advil before they take the vaccine? Because you reduce the inflammation in them. It'll be more effective. Now Emory is now using that to pre-vaccination. So if you reduce the inflammation in these patients, that's how effective it is. Right? In fact, a newspaper article covered this. It said, why is flu shot less effective for the elderly? So we covered it. In fact, in the New York Times, I had a small article on this in terms of why this is the case. The very last thing I want to leave you with is that you think all of this is for science and engineering. No. Beethoven died in 1827, and he had 40 sketches of a 10th unfinished symphony. It was unfinished. So a team of experts in machine learning and musicology took everything they knew about Beethoven, and they fed it into this learning system and asked it to reconstruct the symphony. So this entire AI-driven symphony was played by Bonn Symphony Orchestra for the first time, and then by BBC. They had a whole panel of experts in Beethoven's music. None of them were able to say that it was not a genuine Beethoven piece. Right? The entire symphony was, I was really thrilled to see that. In fact, okay, I don't know if I can play it for you, or at least I don't think it'll let me do it. Sorry. Your network doesn't allow me to do that. But that's okay, you can listen to it. You can go to Google search and do 10th Symphony of Beethoven. And you can listen. It's a canonical Beethoven. When I showed it to somebody who has never, knew what I was playing, I said, that's Beethoven. And that was the 10th unfinished Symphony of Beethoven, done completely by AI. So I want to leave you with the last thoughts. Where's the future going? Looking to the future. In our lab and my viewpoint, large learning models, ChatGPT being an example, is a great example. We're going to go into graphs, knowledge graphs. This is where the future is going, where you're going to connect everything and talk about what the knowledge represents and This is like having Google search on steroids. And so this is where we are going. At least we are trying to convert a lot of biological knowledge into knowledge graphs, ontologies and knowledge graphs, which will help in terms of interpreting where we are going. Imagine going to a doctor and the physician gets it. Imagine this is your physician gets neuropathy due to type 1 diabetes. The physician will be able to see all of this from the measurements and say that, okay, this is the drug I want to give you. Right? This is really not, again, science fiction. This is going to be the future. So we would like to really drive towards the future. For the next, however long I'm going to do research, I'm going to work on 2 areas, Alzheimer's and cancer research. We want to come up with finite solutions using these types of approaches. So AI is a tool, like everything else, and learning is a tool. How we use it is going to define our future. And I'm really excited and looking forward to where we are going with this. And in my lab today, we have a group of people. I have a large lab, 35 to 40 people, and so some parts of them are now working on generating AI and where to go towards the future. So this is where we are. Thank you again for your attention. Please ask me questions or give me ideas, and I'll be more than happy to implement. Thank you very much. And I followed Dennis's instructions. He said 15 minutes, and I finished in 15 minutes.
So, first, thank you for your talk. It's pretty interesting. I don't know if you're, hello. So I had a question thinking about, when you're talking about the future, in terms of how to handle issues related to data representation. So in particular, in your space, working, thinking about health and biology, clinical trials, who we're getting samples from is going to be a very biased sampling process. So how to handle that in terms of thinking about the knowledge that's being gained and generated.
Thank you. Yeah, you have 2 questions embedded in that. So the first question is the question of data representation. The second question of how to get ethical ways of representing data in terms of not having biases induced by, for example, only Caucasian population or only Asian population. They are 2 distinct and interesting questions. The first question is the one which is foundation in this area called ontologies. We cannot represent data unless we have an ontological idea. The basic notion of ontology is words and how the words connect to each other, meaning of words and connections between words. That's what ontological representation is. If you don't have a good semantics structure and good ontological basis, you cannot represent data very well. It's like having all kinds of, it's like if you go to a Sears Roebuck company or Costco, you're going to find shelves which have got a given type of goods, and so therefore you're able to even find the details. So that's based on ontological representation of goods. So we need to have good defined ontologies in the clinical space. One of the projects that I'm advising NIH on is in building these ontological representations. We have got what's now called the minimal clinical metadata model. This minimal clinical metadata model allows us to define diseases in the space of what would represent it in a paper. The second question is infinitely more complex because all measurements, if you look at data, large amounts of data, for example, it's a very good example of that is UK Biobank. UK Biobank has measured sequence 1 billion people. Now guess what fraction of people are Caucasian descent in that 1 million? 98%, or Saxon English essentially. And so there are 1.2% Asians and then smattering of other races. So is what's good for the goose good for the gander? The answer is no. So how do we deal with this issue? This is a very complex issue. Again, National Institutes of Health is struggling with this issue. The only way it's going to happen is if the commodity of measurements, the price of measurements is going to go so low that it can be done in multiple ethnic populations with with ease. That's when we are going to have egalitarian ways of looking at data. Till that time, we can only hope that what's good for the goose is good for the gander. So I hope I've answered your question. It's a genuine problem. It's not a simple problem. Please.
Jim.
So I have to ask this question, given my background as a public sector. And it sort of ties into what Dennis said about the tail of the elephant being lost in the big head and ears swaying around. So I noticed in your roadmap you had, when you talked about uncertainty, it was always like a statistical, stochastic, Bayesian, those were the words that were on there. But they tend to be more susceptible to losing those rare but important issues, and it also ties into, where you were saying if you only have 1% of the population, that has a particular characteristic, you could lose that in the analysis. So what do you see for the idea of being able to handle these situations where you're dealing with huge amounts of data and large dimensionality in losing out those small important points? And I have my own idea, but of course.
Yeah, Jim, it's a beautiful question and very important question. So when I talked, and I didn't elaborate on this in great detail, feature space, I really was referring to the fact that can we, for example, use our feature space knowledge to bring it down to really asking the question about this tail of the elephant as opposed to, because if I know the feature space or the structure of data, I can say that I'm really not interested in looking at 80% or 90% of this data. I'm only interested in looking at small portion. In fact, in the shape and topology of data, we could never discover this luminal B subtype of cancer because people really did measurements on certain things and they said this is luminal B. Or had to. The way we could do it was the shape of the topology of the data told us that this is sticking out as being distinct. And it's a small number of patients, small portion. It is not that it covered a large number of patients. So to me, the future is in terms of looking at more in depth in terms of the feature space, including the shape and topology of data. This is where I think it's going to many other, I'm sure there's going to be more solutions as we go along in the future. But this is where we are with our limited knowledge. Michelle, you had a question.
So it's a philosophical question.
It's a bit of a philosophical question. So I'm a bit of a Freakonomics addict, and the podcast and books, and I listened to one where it talked about machine learning and AI training, an approach to sort of test doctors' diagnoses. So they had a team of doctors, they took a bunch of data, big data set, did all the reductionists, came up with a model, and that worked better when they tested it against the doctors, right? So, in my life I've always really valued expertise, right? And so doctors have expertise, and so just philosophically, I'm just interested in your thoughts on that. Like you have a theme here of context, right? AI is a tool, and it has to be done with context. It has to be constraint learning, structured learning. I don't know, like, are doctors going to become irrelevant? Maybe.
Oh,
No. But explain that to me a little.
Okay. So when you do any learning, you want to have what's called ground truth. Now, what's ground truth? Nobody knows what's ground truth. You're hoping that doctors who have vast amounts of experience are closer to ground truth than, for example, a learning system which does not have all this training and experience. However, the doctor can easily miss something like this thing, the tail you're talking about, because of the fact that the doctor has never seen that particular thing before. I'll give you a real life example. We had a case, I had a case of a parent who came to me once, and this person traveled from San Francisco, and the only reason he came to talk to me was because we are doing something to do with mutations and particular disease. His child had a mutation which gave rise to, it was fatal, in a few years. The only 3 people in the entire universe, entire world, who had those mutations. How do you deal with that? And when they went to a doctor, the doctor said it's autism, it is this or that. They tried all possible standard things, and eventually the child died. But so this is really, I mean, this case was the fact that the doctors missed this, even though they had past experience, because of this unique nature of these patients. And the parent worked really hard to discover this, right? And so I'm giving you an example of where a physician, for majority of the cases, we look at a distribution. For the mean of the distribution, first standard deviation, second standard deviation, the doctor is the ground truth. For the tails of the distribution, the doctor is not the ground truth. Right? This is where it can come in. The more data we learn, this doctors will become more and more. And so philosophically, I don't disagree with you. However, I think assisting the doctor towards diagnosis is where I'm going towards. I'm not saying you replace the doctor, but you're assisting the doctor with diagnosis. And then the area where you didn't touch upon is contextual therapeutics. We do a lot of work in this area. For example, we recently showed that you cannot, I mean, if you take breast cancer, triple negative breast cancer, 30% of patients who are treated with checkpoint immune therapy, which costs thousands and thousands and thousands of dollars, $100,000 or something like that. Only 30% respond. The rest of them not only don't respond, they've got, I mean, worse side effects. So we now have markers for saying which human being, which woman who's got triple negative breast cancer should be treated with checkpoint therapy and who should not get checkpoint therapy. Right? So that's where I'm going towards thinking about how do we contextualize this knowledge towards therapeutics in the future. I hope that answers part of it. It's an important question and there's no answer to it.
In the last few slides of your talk, it seems.
Thank you for the great talk, by the way. In the last few slides, it seems that these methods and these approaches will be integrated towards personalization of medicine and patient-specific planning, ultimately. So what about the confidentiality of the data? If assuming we develop this fancy model that's going to read genomics and genetic information in addition to medical history and all that stuff, what's going to happen in terms of confidentiality?
And this is, again, a beautiful question that we struggle with, that we struggle with all the time. For example, most of the human work we do, we have IRBs, which really, are so stringent and so restrictive. I cannot release any data that is genetic or genomic or even transcriptomic because they say it can be specific to you. The problem is not the confidentiality based on the data. The problem is the societal nature of insurance companies and the way we deal with these things. For example, tomorrow, insurance companies don't care about what diseases you had or what you had, or there's no stigmatization associated with data. That's a societal problem, which is no solution in the science per se, other than the fact we can introduce various types of encrypting and security surrounding the methods. Some of the AI methods today in the healthcare industry, for example, using electronic healthcare, EHR, electronic health records, they are trying to innovate by bringing in some kind of an encryption security into the picture. Still, you'll be able to access everybody's data, but you will never be able to identify the person's data. So they're trying to do that, but it's not successful. If you have a very rare disease mutation, then if you have essentially, if you have sickle cell anemia, it's okay because there are, a lot of percent of the population has sickle cell anemia. But if you have very rare mutation like this child we talked about, it would be really possible to miss who it is to some level. So I don't have an answer for you, except that I think we are going towards a situation where society has to intrinsically think about the fact that these are data points, biology-driven points, and it is not to stigmatize anybody. It's not any different from behavior. Your child goes to school and is a little bit aggressive, then they come back and say ADD, and you have to give some progress. Okay, this is nonsense. It doesn't make sense. It's a natural tendency. You don't look at the circumstances surrounding these things. So it's a societal issue which we have to resolve. I'm absolutely incompetent to do anything at that level. I can only tell you how to deal with the data. Thank you for the question.
With that, let's thank Shankar.